Welcome to Insulin. Today we are going to learn about structure of insulin. So this green segment is called signal sequence. This blue segment is called A chain. This red is called B chain, and this C is C chain or connector peptide. This whole structure is the precursor molecule of insulin, which is called pre-proinsulin. Now, what happened is this signal segment, is digested away in rough endoplasmic reticulum by the enzyme endopeptidase. So now we have B chain, A chain and C chain. And these green lines are representing disulfide bond, and this one represent intra-chain disulfide bond as it is present only in the A chain. The structure without signal sequence is called proinsulin. Now, C chain is also digested away by the enzyme endopeptidase, but this time in Golgi apparatus. Now we have B and A chain connected with disulfide bond and A chain with intra-chain disulfide bond, and with the left out C peptide. This is called biological active insulin or mature insulin. Right, B chain has 30 amino acid and A chain has 21 amino acid. Total we have 51 amino acid in the structure of insulin. Now we will see synthesis of insulin. So here we have the beta cell of the pancreas, and the synthesis starts from the short arm of 11th chromosome. The sequence of the insulin present in the 11th chromosome get transcripted, and mRNA made from this transcription goes to any of the free ribosome present in the cytosol, and the second step is translation. Ribosomes start translation and make a peptide chain, and this peptide chain is none other than signal sequence, which consists of hydrophobic amino acid, and it means it can be dissolved into lipids. So now this peptide chain goes to the rough endoplasmic reticulum along with ribosome and mRNA taking with it. Now further translation is done in the rough endoplasmic reticulum itself, and pre-proinsulin is formed, and this endopeptidase enzyme will act on this pre-proinsulin and make it a proinsulin. This proinsulin will bud out, and this bud will fuse in the Golgi apparatus, and this endopeptidase which is present in Golgi apparatus will digest C peptide connection, and insulin or biological active insulin is formed, which will be bud out as secretory vesicles in association with zinc. Now we will see some important points. Endopeptidase from Golgi complex or Golgi apparatus also works in the secretory vesicles. So if some proinsulin has bud out, can be converted to mature insulin. And insulin is stored in the secretory vesicle with zinc molecules in it.